taken an over-the-counter medication for heartburn? How about an antibiotic for an ear infection? At some point, pretty much all of us have visited a pharmacy to pick up a drug, but we likely don't consider where those drugs come from or how they're made. Whether you're talking about something for seasonal allergies or your grandparents' arthritis medication, the act of bringing a drug to market is long and complex. I'm not an expert, but healthcare triage intern Rachel Hoffman is, and with her help, that's the topic of this week's Healthcare Triage. Before you start making a drug, you need to make sure that there's a target for that drug. Drug targets can include things like cancer cells, viruses, enzymes, or receptors on the surface of our own cells. For example, drugs like morphine and hydrocodone interact with opioid receptors in the brain to decrease pain perception. Once a target has been identified, researchers can begin developing a new drug. New drugs can be discovered in any number of ways. Many of the drugs we use today are derived from natural products. Pyrethrin, a natural pesticide found in some lice shampoos, comes from the chrysanthemum flower, and exenatide, a drug used in treating type 2 diabetes, is a synthetic derivative of a substance found in Gila monster saliva. Drugs can also be developed in the lab to have specific chemical structures to fit a given target, similar to how a locksmith would design a key to fit a lock. This is called de novo drug design, meaning that the drug molecule is a new structure never before seen in nature. The most common way new drugs are discovered today is by a process known as high throughput screening, or HTS. HTS utilizes robotics and high-speed computers to evaluate huge numbers of potential drug molecules quickly and efficiently. If a compound is found to be active for a given target, it may be considered for further investigation. Using any of these methods, an estimated 5,000 to 10,000 molecules may be identified for potential use at an established drug target. Once identified, potential drug compounds undergo rigorous preclinical studies. Researchers will use living cells, animals, and computer models to assess things like how a compound is absorbed, where it distributes throughout the body, how and where it's metabolized, and how the potential drug compound is excreted from the body. The study of drug absorption, distribution, metabolism, and excretion is known as pharmacokinetics, a fancy schmancy word that describes what the body does to a drug. And again, these tests are extensive. In the United States, the Food and Drug Administration, or FDA, requires that such studies be done for every compound before they can be tested in humans. These studies are critical for estimating safety and an appropriate starting dose for human trials. A large percentage of potential compounds are eliminated before and during this stage of drug development. The 5,000 to 10,000 compounds they may have started with will likely be reduced to 5 or fewer candidate drugs. Before a drug can be tested in humans in the United States, an investigational new drug, or IND application, must be filed with the FDA. This application includes a comprehensive report on all the data on animal studies, the chemical compositions of the drug's pharmacokinetic information and side effect profile, and a detailed account of the study design of the forthcoming clinical trials. The FDA carefully reviews these applications before allowing trials to proceed, and it has the authority to halt trials at any time. This type of process in general varies from country to country. The European Medicines Agency, the PDMA of Japan, and Health Canada all have their own regulations and procedures regarding drug trials. Once allowed to proceed, clinical trials progress in 3 phases. In phase 1 trials, researchers establish the safety of a drug by evaluating the pharmacokinetic profile and documenting any and all side effects. Dose escalation may also occur to establish the highest tolerated dose of the compound. Phase 1 trials usually involve roughly 20 to 100 healthy volunteers. An exception to this are trials investigating cancer drugs, in which seriously ill patients who are not responding to standard therapy are the study population. In phase 2, the candidate drug is tested to establish safety and efficacy of the compound. Such tests are usually done in about 100 to 600 volunteers with the disease state under investigation. Pharmacokinetics and side effects are still monitored, but researchers are primarily interested in understanding if the drug has the effect we want on disease and its symptoms. Phase 2 inquiries also focus on finding the optimal dose for sick patients. Phase 3 is very similar to Phase 2 in that its purpose is to establish safety and efficacy, but it's done on a much larger scale. Thousands of volunteers with the disease state from all over the world are involved in these trials. The larger numbers allow researchers to present statistically significant data to the FDA about the drug's safety and efficacy. Information from phase 3 is also key in establishing the labeling data that will accompany the final drug product. You know those thin folded sheets of paper that come with your prescriptions that look like a Dickens novel printed in 2-point type? All of that stuff was figured out and finalized in phase 3 clinical trials. Okay, so after all this should all go well, it's time to reassess. The drug is safe, the drug is effective. Now we have a new drug ready to be prescribed, right? No. Everything needs to be approved by a nation's regulatory body once again before the drug can be marketed. Throughout all the phases of clinical trials, investigators are constantly analyzing their data and compiling information for submission to regulatory bodies. In the United States, at this point, a new drug application, or NDA, must be submitted to the FDA. The FDA then reviews the data and decides whether or not to approve the drug. Submission of an NDA does not guarantee approval. The FDA may issue an approvable letter to the investigators requiring additional studies, or they may deny approval of the drug altogether. But let's say the drug is approved. Then what? Well, naturally the drug's gonna work perfectly. Nobody experiences any nausea or has dizziness when standing. We all live forever, and Ned Stark turns out not to be dead after all, right? Guys? No, of course not. Even though a drug may have been studied over decades and thousands of volunteers, it's impossible to know exactly how this drug will affect every single patient. That's why there's a fourth phase of drug research known as post-market surveillance. In post-market surveillance, drugs are continually monitored, and pharmaceutical companies often submit long-term safety data to the FDA. Consumers and practitioners can also participate in phase 4 by reporting adverse drug events to the FDA's Med.USA program. So, to recap, the drug development process is complicated. You find a target, test for a compound to hit it, do preclinical studies, get approval for human trials, complete phase 1, phase 2, and phase 3, then get approval again, and finally monitor the drug's use until forever to make sure it's still safe and working. This whole process of bringing a drug to market takes a really long time. From a drug's discovery to the point where you can finally find it in the pharmacy takes an average of 15 years according to industry. Let's put that into perspective. 15 years ago, Harry Potter and the Goblet of Fire was new on the shelves, the PS2 had just come out, and the only Marvel movie in theaters was X-Men. The original X-Men. Even then, we sometimes screw up. Some people claim it's too easy to get a drug approved. Others claim it's too hard. Debate should continue, and it will. Healthcare Triage is supported in part by viewers like you through Patreon.com, a service that allows you to support the show through a monthly donation. We'd especially like to thank our honorary research associates, Cameron Alexander and Kadim Salah Mohammed. Thanks, Cameron and Kadim. Healthcare Triage will always be free to watch and share, but your support helps make it bigger and better. Taken an over-the-counter medication for heartburn? How about an antibiotic for an ear infection? At some point, pretty much all of us have visited a pharmacy to pick up a drug, but we likely don't consider where those drugs come from or how they're made. Whether you're talking about something for seasonal allergies or your grandparents' arthritis medication, the act of bringing a drug to market is long and complex. I'm not an expert, but healthcare triage intern Rachel Hoffman is, and with her help, that's the topic of this week's Healthcare Triage. Before you start making a drug, you need to make sure that there's a target for that drug. Drug targets can include things like cancer cells, viruses, enzymes, or receptors on the surface of our own cells. For example, drugs like morphine and hydrocodone interact with opioid receptors in the brain to decrease pain perception. Once a target has been identified, researchers can begin developing a new drug. New drugs can be discovered in any number of ways. Many of the drugs we use today are derived from natural products. Pyrethrin, a natural pesticide found in some lice shampoos, comes from the chrysanthemum flower, and exenatide, a drug used in treating type 2 diabetes, is a synthetic derivative of a substance found in Gila monster saliva. Drugs can also be developed in the lab to have specific chemical structures to fit a given target, similar to how a locksmith would design a key to fit a lock. This is called de novo drug design, meaning that the drug molecule is a new structure never before seen in nature. The most common way new drugs are discovered today is by a process known as high throughput screening, or HTS. HTS utilizes robotics and high-speed computers to evaluate huge numbers of potential drug molecules quickly and efficiently. If a compound is found to be active for a given target, it may be considered for further investigation. Using any of these methods, an estimated 5,000 to 10,000 molecules may be identified for potential use at an established drug target. Once identified, potential drug compounds undergo rigorous preclinical studies. Researchers will use living cells, animals, and computer models to assess things like how a compound compound is absorbed, where it distributes throughout the body, how and where it's metabolized, and how the potential drug compound is excreted from the body. The study of drug absorption, distribution, metabolism, and excretion is known as pharmacokinetics, a fancy schmancy word that describes what the body does to a drug. And again, these tests are extensive. In the United States, the Food and Drug Administration, or FDA, requires that such studies be done for every compound before they can be tested in humans. These studies are critical for estimating safety and an appropriate starting dose for human trials. A large percentage of potential compounds are eliminated before and during this stage of drug development. The 5,000 to 10,000 compounds they may have started with will likely be reduced to 5 or fewer candidate drugs. Before a drug can be tested in humans in the United States, an investigational new drug, or IND application, must be filed with the FDA. This application includes a comprehensive report on all the data on animal studies, the chemical compositions of the drug's pharmacokinetic information and side effect profile, and a detailed account of the study design of the forthcoming clinical trials. The FDA carefully reviews these applications before allowing trials to proceed, and it has the authority to halt trials at any time. This type of process in general varies from country to country. The European Medicines Agency, the PDMA of Japan, and Health Canada all have their own regulations and procedures regarding drug trials. Once allowed to proceed, clinical trials progress in 3 phases. In phase 1 trials, researchers establish the safety of a drug by evaluating the pharmacokinetic profile and documenting any and all side effects. Dose escalation may also occur to establish the highest tolerated dose of the compound. Phase 1 trials usually involve roughly 20 to 100 healthy volunteers. An exception to this are trials investigating cancer drugs, in which seriously ill patients who are not responding to standard therapy are the study population. In phase 2, the candidate drug is tested to establish safety and efficacy of the compound. Such tests are usually done in about 100 to 600 volunteers with the disease state under investigation. Pharmacokinetics and side effects are still monitored, but researchers are primarily interested in understanding if the drug has the effect we want on disease and its symptoms. Phase 2 inquiries also focus on finding the optimal dose for sick patients. Phase 3 is very similar to Phase 2 in that its purpose is to establish safety and efficacy, but it's done on a much larger scale. Thousands of volunteers with the disease state from all over the world are involved in these trials. The larger numbers allow researchers to present statistically significant data to the FDA about the drug's safety and efficacy. Information from Phase 3 is also key in establishing the labeling data that will accompany the final drug product. You know those thin folded sheets of paper that come with your prescriptions that look like a Dickens novel printed in 2-point type? All of that stuff was figured out and finalized in Phase 3 clinical trials. Okay, so after all this, should all go well, it's time to reassess. The drug is safe. The drug is effective. Now we have a new drug ready to be prescribed, right? No. Everything needs to be approved by a nation's regulatory body once again before the drug can be marketed. Throughout all the phases of clinical trials, investigators are constantly analyzing their data and compiling information for submission to regulatory bodies. In the United States, at this point, a new drug application, or NDA, must be submitted to the FDA. The FDA then reviews the data and decides whether or not to approve the drug. Submission of an NDA does not guarantee approval. The FDA may issue an approvable letter to the investigators requiring additional studies, or they may deny approval of the drug altogether. But let's say the drug is approved. Then what? Well, naturally the drug's gonna work perfectly. Nobody experiences any nausea or has dizziness when standing. We all live forever, and Ned Stark turns out not to be dead after all, right? Guys? No, of course not. Even though a drug may have been studied over decades and thousands of volunteers, it's impossible to know exactly how this drug will affect every single patient. That's why there's a 4th phase of drug research known as post-market surveillance. In post-market surveillance, drugs are continually monitored, and pharmaceutical companies often submit long-term safety data to the FDA. Consumers and practitioners can also participate in Phase 4 by reporting adverse drug events to the FDA's Med.USA program. So, to recap, the drug development process is complicated. You find a target, test for a compound to hit it, do preclinical studies, get approval for human trials, complete Phase 1, Phase 2, and Phase 3, then get approval again, and finally monitor the drug's use until forever to make sure it's still safe and working. This whole process of bringing a drug to market takes a really long time. From a drug's discovery to the point where you can finally find it in the pharmacy takes an average of 15 years according to industry. Let's put that into perspective. 15 years ago, Harry Potter and the Goblet of Fire was new on the shelves, the PS2 had just come out, and the only Marvel movie in theaters was X-Men. the original X-Men. Even then, we sometimes screw up. Some people claim it's too easy to get a drug approved. Others claim it's too hard. Debate should continue, and it will. Healthcare Triage is supported in part by viewers like you through Patreon.com, a service that allows you to support the show through a monthly donation. We'd especially like to thank our honorary research associates, Cameron Alexander and Kadim Sa.
Very good morning to all of you. I myself, Y. Ratna Sindhu, Assistant Professor, Department of Pharmaceutical Regulatory Affairs, Hindu College of Pharmacy. A very good morning to one and all. On behalf of Hindu College of Pharmacy, we, the Department of Pharmaceutical Regulatory Affairs, welcomes you for this fabulous online program, PRAJWALA, an international webinar series on contemporary issues in pharmaceutical regulatory affairs. Ours is one of such institutions, Hindu College of Pharmacy, started in 1999 and educating students to face the real world in all possible ways. Since esteemed institute stands for its excellence, and there are many people to mention for their marvelous service to uphold the policy of educating and benefiting the students with knowledge in Hindu College of Pharmacy. They are none other than our management. Sri Jupudi Rangaraj Garu, Chairman, and Dr. S. Narasimhan Rao Garu, Secretary and Correspondent, and our erstwhile founder, Secretary, and later Chairman, presently our respected Honorary Chairman, PCA member, Dr. M. Radhakrishnamurthy Garu, who built the institution brick by brick, and our Principal, Sir, R. Govindarajan Garu. I am glad to introduce them to you all. We are also proud to say about our Department of Pharmaceutical Regulatory Affairs. He is led by dynamic head of the department, Dr. M.V. Nagabhushanam Garu. Under his leadership, this department has developed like an educational hub. We are proud to say it is one of the best departments in Hindu College of Pharmacy. Now it's time to welcome our speaker for today's session, Dr. Siva Reddy Challa, Research Collaborator, Department of Cancer Biology and Pharmacology, University of Illinois College of Medicine at Peoria, USA. Sir will deliver his talk on today's session, latest regulatory guidelines for clinical research in BA/BE studies and good clinical practice. Finally, it is more important to welcome you all. Finally, it is very important to welcome you all with a deep gratitude for your fabulous responses for the program organizing by Department of Pharmaceutical Regulatory Affairs. Now we will start the program with a prayer song.
कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युत शङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
Now I request our HOD, convener of today's program, Dr. M.V. Nagabhushanam Garu, to start the session with his opening remarks. Over to our respected HOD, sir.
Good morning to all esteemed participants present now to this fourth day of International Webinar Series PRAJWALA, organized by Department of Pharmaceutical Regulatory Affairs, Hindu College of Pharmacy, Guntur, Andhra Pradesh. From 25th June to 28th June 2020, we are organizing this series of webinars on contemporary issues of pharmaceutical regulatory affairs. I feel happy to announce that more than 3,000 participants have registered across the globe from the fraternity of pharmacy to this international webinar. Among the registered pharmacists participants from various countries, there are 487 members, doctorates, 81 are professors, 257 are associate professors, and 688 are assistant professors. Registrations were received from USA, Malaysia, Australia, Ireland, Ethiopia, Saudi Arabia, and many other states of India. Among them, nearly 50% of the respondents are employees and more than 1,500 are students of B.Pharm, M.Pharm, and PharmD programs. We are happy to receive this kind of fabulous response all over the world. And generic drugs is the one which is playing a significant role in this modern healthcare providings of the life, which are effective and low-cost alternatives to the corresponding brand-named drugs. Indeed, the profile of generic drugs has only increased in recent years as public and political pressure across the ideological spectrum has mounted to help combat out of control drug prices. In response to these regulatory agencies which are facilitating the development and approval of new generic drugs, instead of a full new drug application as required for new drugs, an abbreviated new drug application, which is known as ANDA, is used for the approval of the generic drugs. Perhaps the most critical requirement for ANDA submission is that applicant has to submit and demonstrate that the proposed generic version of the drug product is bioequivalent to that of reference listed drug product, which in short we call it as RLD. FDA approvals or any other generic drug approval should consider several factors while planning their bioequivalence studies, careful considerations of appropriate study designs, sampling schedules, desired release characteristics, and those many other issues for designing a successful BA/BE development program. Careful consideration of appropriate study design, sampling schedule, all these are very much essential for successful development of BA/BE study program. The key success is to appreciate how people learn, understand the thought process that goes into the instructional design, what works well, and a range of different ways of achieving the goals. To make these unexpected days more useful and fruitful due to the prevalence of the pandemic COVID-19, our Hindu College of Pharmacy Management has guided our teachers to integrate this webinar technology seamlessly into the curriculum instead of viewing it as an add-on or as an event. On this behalf, I, Professor M.V. Nagabhushanam, Head of the Department of Pharmaceutical Regulatory Affairs, extends heartiest welcome to a humanitarian, generous, and philanthropist, a renowned advocate of Guntur city, and an able administrator, successful leader, and moreover, our beloved chairman, Sri Jupudi Rangaraj Garu, to this webinar series. And also, I cordially welcome a well-known physician, visionary, insightful, and inspiring person, Dr. S. Madhusudan Ragaru, Secretary and Correspondent of Hindu College of Pharmacy. I extend my ardent welcome to our Principal, Dr. Govindarajan, Vice Principal Sunita Madam, PharmD Director Sitaramaya Garu, to this international webinar series. Have learned that people will forget what you said, people will forget what you did, but people will never forget how you made them feel. To make us feel about his knowledge, about the BA/BE studies especially, today we have with us Dr. Siva Reddy Challa, a research collaborator from Department of Cancer Biology and Pharmacology, University, Illinois College of Medicine at Peoria, USA, who has accepted our request and invitation to spare his time and share his knowledge to enlighten all of us on the topic, latest regulatory guidelines for clinical research in BA/BE studies and good clinical practices. I heartfully invite Dr. Siva Reddy Challa for this webinar session. I offer a warm welcome to all faculty members, professionals from pharma industry, students from India and abroad who have constantly showered their love and support and their benign cooperation throughout this program for the grand success. Thank you. Now I request our department senior faculty member, D. Nagarjun Reddy Garu, to introduce our today's speaker, Challa Siva Reddy Garu, Dr. Challa Siva Reddy Garu. I request
morning, sir. Good morning, one and all, those who have attended this session. It's my pleasure from Department of Pharmaceutical Regulatory Affairs and from the College of Pharmacy to brief about the biodata of Dr. Siva Reddy Challa garu, who is research collaborator, Department of Cancer Biology and Pharmacology, University of Illinois College of Medicine and Area, Illinois, USA. Sir is going to deliver his speech on latest regulatory guidelines for clinical practice, research in bioavailability and bioequivalence studies, and good clinical practices. Dr. Siva Reddy Challa has done his M.Pharm and PhD. He is working presently as a research collaborator, Department of Cancer Biology and Pharmacology, University of Illinois College of Medicine at Korea. Sir has completed his M.Pharm Pharmacology from Andhra University, Visakhapatnam, Andhra Pradesh. Sir has completed his PhD in cardioprotective actions of quercetin and routine ischemia riboflavin induced myocardial infarction. Sir is presently offering his services as research collaborator on an NIH-funded project on post-stroke brain damage. Sir is having his experience as clinical research associate CRA at BioServe Clinical Research Private Limited and fulfilled responsibilities in clinical protocol preparation, clinical study monitoring, informed consent form, presentation to volunteers, and preparation of clinical study reports. Sir also offered his services as senior research officer at Glenmark Pharmaceuticals Limited, Mumbai, India. To his credit, Dr. Siva Reddy sir is having 32 research publications, peer-reviewed international scientific journals. Sir is acting as peer reviewer for many scientific journals of international repute. Acted as a mentor for many students for PharmD and PhD. Sir is having his expertise in preparation of clinical study reports for submission to international regulatory agencies. And to his honor awards and achievements, Sir has achieved All India rank 125 in GATE and also research assistantship. Sir has received Chokopalli Pichaya Memorial Best Teacher Award 2015 and many. Sir, it's our pleasure to brief your biodata. And thank you, sir. Thank you for giving this opportunity to our expert. Over to Siva Reddy Challa.
I request Dr. Siva Reddy Challa to start this session.
Okay, sir.
Welcome.
Sir, okay, before I proceed, I would like to keep annotations in mute. So, could you please guide me how to do that? Sir, unmute yourself, sir. Look at this. So I'm okay. Sir, Dr. Nagakrishna, sir. Could you please unmute yourself? Okay.
Yeah, unmuted. Okay. And I'm informing to my faculty member.
Okay, sir.
To go for annotations. To
Okay.
give you the annotations.
Sir, good morning, sir.
Siva Reddy garu.
Yeah. Good morning, sir.
Good morning, sir. Sir, please share the screen.
Okay.
Then go for more options.
Sir, are you able to see? Are
Yes,
you
sir.
able to see
Yes,
the
sir.
presentation?
Yes,
Yeah.
sir. Go for more options.
More options. Okay.
Then disable participant annotations.
Okay, 1 minute, sir. Okay. Okay, more options. Okay, I did it, sir. Okay.
Okay, sir. Now you can start the session, sir.
Okay.
Yeah.
Okay, thank you, Dr. Nagarjuna Reddy garu, for introducing me. So I am quite happy to see to various faculty members and students of the various institutes. So I am proud to say that I have served for some time in Hindu College of Pharmacy as a faculty member. and also I am an humble student of Dr. Nagabhushna, MV Nagabhushna garu. So I always admire him as a teacher. Okay, it is a great opportunity for me to deliver a lecture. So good morning, everyone. And I am first of all, I am grateful to the management of Hindu College of Pharmacy, especially Secretary Garu and Chairman Garu, Sri Rangaraj Garu, and Secretary Dr. Madhusudan Rao Garu, and the principal Dr. Govindarajan Garu, and Vice Principal Dr. Sunita Madam, and okay, and finally I would like to thank the audience and many faculty members and many students from the various institutes. So good morning, one and all. So now I am going to present my title of the presentation is like latest regulatory requirements of VAB studies and good clinical practice. So the topic which is assigned me based on my experience in clinical research in BioServe Clinical Research Private Limited. So where I used to write the protocol preparation. I used to write protocols and I used to monitor the clinical research, bioequivalence studies, and I used to participate in informed consent form, in taking the informed consent from the healthy volunteers. So I actually participated for some time in clinical research. So where I know mainly the guidelines of good clinical practice. So in addition to GCP guidelines, I would like to talk more about regulatory requirements of VAB studies. So most recently in India, the guidelines have been changed, particularly in Schedule Y of the Drugs and Cosmetic Act has introduced so many new rules and procedures have been changed. So I would like to give emphasis on the certain things which are modified in the latest regulations. So let me move on the presentation. So before going to the discuss, we should learn to know the types of or the different types of regulatory mechanisms. So here they define very clearly what is law and what is regulation and what is guideline. So here when we see the differences, law is a rule of conduct enforced by a controlling authority. So it's like Drugs and Cosmetic Act is one of the example for law. And whereas regulation is nothing but like one best example is Schedule Y of the Drugs and Cosmetic Act. Actually, Schedule Y is represented as a regulation, whereas the Drugs and Cosmetic Act is a law. So we have to implement this, to follow and to maintain these regulations. We need to have certain guidelines. So that is the reason the Indian government has provided the ICMR guidelines, and we have Indian GCP guidelines, and internationally we have ICH GCP guidelines, and many more guidelines all over the world. Like in European countries, we have European Medicine Agency guidelines. Depending on the various country, the guidelines are varying. Like Canadian, for Canada, we have separate guidelines. So when do we do bioequivalence studies? So usually we conduct the bioequivalence studies for generic formulations. So, but there are other instances where we conduct bioequivalence studies, like here. So, like when sometimes one pharmaceutical industry, they want to change the formulation from capsules to tablet. So in case of change of formulations, they can go for bioequivalence studies. So similarly, sometimes we see the change of process or manufacturing site. So here, whenever there are changes expected in formulations, So there we need to conduct the bioequivalence studies. So let us first of all know what are the various differences between brand name drugs and generic name drugs. So many people know about all these basic things. Still, we need to know basic things. Okay, the brand name drugs. When you compare, it is also called as innovative drugs. So whereas in case of generic name drugs, they are considered as copies of brand name drugs. So in case of brand name drugs, they are initially marketed as new chemical entities. So we know for brand name drugs, we need to apply for new drug application NDA. So similarly, it is supplied by only one drug company, that is called innovator company. So it is sold under the company's trademark name. Trademark is it has a particular brand name, and the cost is more in case of branded drugs. So considering the more healthcare cost on the healthcare burden and healthcare cost, so many regulations came to introduce the generic drugs into the market. Here, when you look at the generic name drugs, okay, they are the copies of brand name drugs and produced after original patent expires. So we all know, okay, the patent expires from the innovative company. The patent of the innovative product expires. So as soon as it expires, okay, many companies, they look forward to apply for ANDA application, abbreviated new drug application, in order to get their generic product approved. So it may be supplied by more than one company and may be sold under active ingredient name. Okay, generally we see generic name drug is just like a active ingredient name. So cost is relatively less when compared to the branded drug. So let us see what is therapeutic equivalence. So we need to know, okay, basically therapeutic equivalence, pharmaceutical equivalence, and bioequivalence. These are the important terms in bioequivalence studies. The FDA classifies as therapeutically equivalent those products that meet the following general criteria. So they are approved as safe and effective. That means they should meet the requirements of bioequivalence. When we say, okay, drug products are bioequivalent, it means that, okay, their safety and efficacy are to be considered equivalent. So we can't say here equal. We are all the time we are using the equivalent word, so that we need to consider. So equivalent means they are not exactly the same. Okay, they have some variation, but the variation may be within the limits. So generally we know, okay, certain many guidelines they will give the variation between the generic product and branded product. It is not more than 20%. Generally we see the confidential 90% confidence interval range. It varies from it varies from 80 to 125%. So when you see 80 to 125%, it looks like a 20% variation is considered. Based on the particular regulatory agency, the confidence interval ranges may vary. So okay, I'm going to discuss all those things in the coming slides. So here they are pharmaceutical equivalents in that they. So what does exactly pharmaceutical equivalent mean? That contain identical amounts of the same active drug ingredient in the same dosage form and with the same route of administration, and meet compendial other applicable standards of strength, quality, purity, and identity. So we can also therapeutic equivalence is considered as a summation of it is a combination of bioequivalence and pharmaceutical equivalence. So here bioequivalence represents that it is we can say that the drug efficacy and safety and efficacy are equivalent to the innovative product. So whereas pharmaceutical equivalence indicates that in both the cases we are using the same dosage and the same route of administration and the same dosage form. Okay, both formulations are more identical in many aspects. So the combination of bioequivalence and pharmaceutical equivalence must result in therapeutic equivalence. So ultimately, one is expecting the therapeutic actions at the target site. So let us see what is the generic drug. The generic drug will be considered to be bioequivalent to a reference listed drug. Here, the rate and extent of absorption of the generic drug doesn't show a significant difference from the rate and extent of absorption of the listed drug. So here, the rate and extent of rate means, okay, we know in terms of pharmacokinetic parameters, it is Tmax. Extent of absorption means it's nothing but AUC, area under the curve. So we most of the time, okay, we consider the pharmacokinetic parameters most commonly Tmax, Cmax, and AUC. Particularly in this bioequivalence studies, we more talk about Cmax and AUC. We are more concerned about the Cmax and AUC. So particularly AUC 0 to T and 0 to infinite. These are more important. So the rate means Tmax and the extent of absorption means it is AUC. The rate and extent of absorption of the generic drug is considered as the bioavailability of generic drug. So it doesn't show a significant difference from the rate and extent of absorption of the listed drug. Listed drug means here, okay, whatever the innovative drug with which we are comparing the generic drug. So doesn't show the significant difference. Okay, we need to statistically prove that there is no significant difference between these two products. So that needs to be done by statistical analysis. So the FDA considers two products bioequivalent if the 90% confidence intervals of the relative mean Cmax or AUC 0 to T or AUC 0 to infinite of the test to the reference. We always take the ratio of the Cmax of the test to the Cmax of the reference, or okay, AUC 0 to T of the test to the reference, okay, to AUC 0 to T of the reference. So we are going to see the relative ratio. Okay, so here we are just seeing the whatever the range we are seeing, that is a relative ratio from the test to the reference. So all the time we should remember. So we are looking at the relative ratio. So okay, when you consider test product, Cmax is in the numerator and reference product Cmax is in the denominator. When they are equal, so yeah, it becomes it is equal to 1. So in terms of percentage, it is 100%. So when the bioavailability of generic product and bond product are equal, that means that okay, then you are going to get the percentage 100%. So based on that, so we are going to see if it is 100%, exactly they are equal. So if it is 80%, maybe 20% variability is there. If it is more than, okay, it is like 120% or 125%. So we have variability of up to 25%, and like this. Okay, let me explain more detail about it. So this is the best example we generally explain in many conditions in bioequivalence. So these are the acceptable limits. So the these acceptable limits will vary based on the regulations of okay, in the US FDA regulations we see most of the time 80% to 125%. At 90%, I am talking about 90% confidence intervals. Most commonly, the confidence intervals which are considered for bioequivalence studies is 90% confidence intervals. So in this case, if you look at this example, so this pink, whatever the normal distribution curve which was shown in pink color, it is innovative product. It is exactly in the middle of the range. So when you look at other formulations, when you look at this formulation, here the upper limit of the confidence interval is within the limit, but lower limit is below the limit here. So it may be either Tmax or AUC. So we generally consider, okay, generally these acceptance limits are represented for Cmax and area under the curve. These are the area under the curve is also represented as a exposure of the drug in the plasma, exposure of the drug in the plasma. That means the amount of drug present in the plasma or blood circulation, systemic circulation. So whereas in other, you see in this example, it is completely the concentrations here it is beyond the upper limit of the confidence interval. That means it is not it's not safe. So we have high concentrations than the innovative product. So that is the reason it is not safe. So whereas you see, look at this formulation. This is below the lower limit of the confidence interval. So below lower limit is 80%. It is below the lower limit. So it is not bioequivalent. Because when compared to innovative product, it is not bioequivalent, and it's it's not going to show the efficacy also, because minimum concentrations required to produce the effect. So it is not going to yield the minimum concentrations required to produce the effect. Okay. So in the coming to in this formulation, there is a lot of variability. So and upper limit is beyond the 125%. So sometimes some formulations they may have high variability. So this graph is a good representation. This is one thing. I have a doubt all the time. Why is 90% confidence interval acceptable range is 80 to 120% instead of 80 to 120%? So when I was studying my MPharm, my professor used to say 20% variation between the innovative product and generic product is acceptable. So when it is 20%, it must be like 80 to 120%. But I was just wondering, how come it is 80 to 120%? So that has made me to search about more literature, and I came to know here. When you look at this ratio, so these are the test AUC values. Like we have 0.8 test product, and the reference is 1. Reference is always we take it as a 1. So when you take the ratio of 0.8 and 1, 0.8 by 1, it is 0.8. So percentage is 80%. So here this is a natural logarithm of this ratio. This is a natural logarithm of this ratio. So when you take the natural logarithm of 0.8, it is minus 0.223. So here, when you apply the natural logarithm for all these ratios, so we are going to get the values like this. So literature says, actually this is following symmetrical and symmetrical normal distribution pattern. So that is the reason they have considered the lower limit is 80% and upper limit is 125%. Because of this natural logarithm values, they present plus and minus both the same values here. And most of the guidelines, most of the regulations, they say the range is 80 to 125%. But sometimes it may vary, like when we have low therapeutic index drugs, when we have the drugs with high variability, particularly in case of low therapeutic index drugs, and in some special cases, they are going to consider the variability plus or minus 30%. Sorry, and it's not the low therapeutic index drugs. Actually the drugs with high variability, so they are going to consider the high variability like plus or minus 30%. So in some cases, even for certain drugs, they accept the acceptable range maybe 70 to 143%. So abbreviated new drug application. So all of us, we know the pharmaceutical companies, they apply for ANDA to get their generic product approved and marketed in the country where they are going to market the drug. Like for example, if any Indian company is ready to launch their generic product in USA, they need to get the approval of US FDA to get the approval and market the product in USA. So later on, when the study is over, the regulatory authorities will scrutinize all the document. Regulatory authorities, sometimes they may visit the CRO where the study was conducted, and they will audit the trial site and clinical research site to scrutinize each and everything. So here, the abbreviated new drug application contains data which when submitted to FDA's CDER. This is Office of Generic Drugs with FDA. So this is called Center for Drugs and Evaluation Research. So this is considered as the Office of Generic Drugs. It provides for the review and ultimate approval of a generic drug product. This is the responsible office where all these approvals are happening in USA. Once it is approved, an applicant may manufacture and market the generic drug product to provide a safe, effective, and low-cost alternative to the public. So all approved products, both innovator and generic, are listed in FDA's approved drug products with therapeutic equivalence evaluations. So all of us know we have a orange book. So where all FDA approved drug products with therapeutic equivalence evaluations are published in the orange book. So this is like a the orange book is very well known for all the bioequivalence centers. So to whatever the drug which is approved, it is going to be published in the orange book. So here a drug product that is comparable to a brand or reference listed drug product is a. So this I just repeated the same point about the generic drug. Okay, it is not necessary. Okay. It termed abbreviated or cut short. Okay, why it is called as abbreviated new drug application? Because they generally not required to include the animal data and the clinical data to establish the safety and efficacy. So particularly in the ANDA approval, okay, for ANDA approval and for the application of ANDA, so we don't have to submit the animal data and the clinical trial data. So that is the reason it is called as a abbreviated new drug application. So basic generic drug requirements are just like I explained earlier. Same active ingredient, same route of administration, and we need to have same dosage form, same strength, same conditions of use. So inactive ingredients already approved in a similar NDA should be used. So ANDA, when you see the evolution process of how ANDA came into the picture, in 1970, FDA established the ANDA as a mechanism for the review and approval of generic versions. So later on, here, before 1978, generic product applicants were required to submit complete safety and efficacy through clinical trials. But before 1978, we can consider here, before 1978 and after 1978, there was a change. So post 1978, applicants were required to submit published reports of such trials, documenting and safety and efficacy. But whereas in 1978, generic product applicants, they are required to submit complete safety and efficacy through clinical trials. So it is a difficult task earlier. So now it has become simple because they don't have to conduct the clinical trials here after 1978. So neither of these approaches was considered satisfactory, and so finally there was an introduction of Hatch-Waxman Act in 1984. So the introduction of Hatch-Waxman Act has changed a lot. Changed the things very differently. So these are the things which are introduced with the Hatchman Act. Restoration of a portion of their intellectual property rights lost during the pharmaceutical approval process. So it obviously takes some time for the generic product to get approved. So in this case, they are going to compensate the time, whatever the approval time that was consumed, that was restored by the regulatory authorities. So and then reduce the average delay between patent expiration and generic entry into the consumer market. So now we have like certain, I think you might have gone through the regulations like paragraph certification 1, paragraph certification 2, like certain paragraph certification 3 and 4. So I'm not going more details about the paragraph certifications. So here, by introducing all these things, so the pharmaceutical companies, they can even make an A&D application before the patent expiration of the innovative drug. So that is the provision they have created with this, reducing the average delay between. One cannot wait till the innovative drug patent expires. So maybe one can apply even before the expiration of the patent of innovative drug. So this has created the provision to apply for the ANDA. So even before patent expires, that provision is created. And here we have 180-day market exclusivity period for the first market generic. So if any company which has made the application, made the generic ANDA application under paragraph 4 certification, so they will be given 180-day market exclusivity period for the first marketed generic product. So this is the one thing they are introduced newly. So under 5-year exclusivity period during which generic manufacturers cannot submit FDA applications. This is a blessing for innovative companies. So this is a 180-day market exclusivity period is a beneficial thing for generic pharmaceutical companies. So the pharmaceutical companies which are producing generic formulations. And 5-year exclusivity period and 3-year exclusivity period. These two are mainly applicable to innovative company. So here 5-year exclusivity period during which generic manufacturers cannot submit the FDA applications. So this period, this is the period where the generic manufacturers, they cannot apply the FDA applications. Okay, this is a beneficial thing for innovative companies. 3-year exclusive period. This is for improved versions of brand pharmaceuticals. So they already have some brand formulation in the market. They have improved the version of the brand pharmaceutical. Okay, in that case, they have again they have been given 3-year. They will be given 3-year exclusive period for improved version of the brand pharmaceuticals. So this is how we see the normal organization of the clinical research or clinical trial. So in clinical trial, we need to register the clinical trial in clinical trial register of India, and sometimes internationally we have clinical trial registry. So in the bioequivalence studies, we don't have to register the bioequivalence studies in CTRI. It is applicable only for clinical trials. So the remaining complete organization is relevant to the bioequivalence studies. Okay, here we have different components like different members. They play a key role in the bioequivalence studies. Sponsor and regulatory authorities, and then CRO, clinical research organization, and we have institutional review board or institutional ethics committee. These are the 4 important components which play an important role. So let me give an example where I worked in BioServe. So BioServe, like it is like a CRO where I worked. So I worked like a CRA, clinical research associate. So I worked under the guidance and under supervision of clinical research coordinator. Okay, so he is superior to me, and CRC is working in collaboration with the clinical investigator. So we have designations like this. The role of CRA is to design the protocols and to take the informed consent form and to monitor the overall bioequivalence study. So these are the functions of the CRA. So we usually conduct all these bioequivalence studies on healthy human volunteers. These are the main important participants. So sponsor has to make an application to conduct the bioequivalence study to the regulatory authority. So like in India, regulatory authorities is CDSCO. This is the Control General of India is going to approve the bioequivalence studies. So before that, they need to approve the protocols, all the protocols from IEC. So that whatever the protocols which are approved by the IEC, they are going to execute it in the CRO. So we have many CROs in India, particularly in Hyderabad. We have like Vimta Labs and Cipra Labs and Arvindo, Trident. So we have so many CROs in Hyderabad. Okay, they are all going to do, many of them, they are going to conduct bioequivalence studies rather than phase one trials. Okay, so I'm going to move on. What are the different designs in bioequivalence studies? So okay, we have so many designs like parallel design, crossover design, non-replicative crossover design. So in case of parallel design, okay, so we need to see here. Okay, these are the subjects. Okay, healthy volunteers. They are randomized. They are randomized and they are divided into two groups. Like okay, I mentioned the 7 subjects and 7 subjects. Generally, in pilot studies, the volunteers, number of volunteers which are taken are 12 to 14 members. So that's the reason I mentioned that most of the studies that I involved are mainly pilot studies. So in case of pilot studies, like we divide the volunteers, healthy volunteers into 2 groups. So one group is going to be subjected to the one particular treatment. So in this case, okay, one proportion of the patient, proportion of the healthy volunteers are will be administered test drug. So whereas another group of subjects they are administered with the reference drug. So we have a sequence 1. Okay, here sequence 1 is only T. So sequence 2 is only R here. So this is called okay, 2 sequence no period design. So we have no periods here. I mean, sorry, we have 1 period but no washout period. So I would like to say there is no washout period. So we have only 1 period. So, but the problem in this study is this study generally conducted for the drugs which are having long elimination half-life. So even when the drug is having elimination half-life more than 24 hours. So it takes around, let us say around 5 to 6 half-life to completely, for the drug to be eliminated completely from the body, it takes 5 to 6 half-lives. So for that reason, okay, the study is going to be very lengthy because even for a single period, it takes around, sometimes the study may run for 10 days. So it is difficult to maintain the maintain the healthy volunteers for such a long period, like 1 week or 10 days. Usually in most of the studies we conduct, in the period 1, the healthy volunteers will spend 3 days, not more than 3 days most of the time. So okay, first day evening they are checked in, and then second day the total study. So they are in-house, and we collect the blood, we use it to collect the blood samples at various time points which are fixed, and then third day they are going to check it out. So this is how okay generally the healthy volunteers also prefers this kind of schedule. So in case of parallel design, it is not practically feasible to maintain the healthy volunteers for more than 7 days or 10 days. It's difficult. So however, so here it is very difficult to run the another period. That is the reason only they run okay. They will conduct the study for only single period. So this is considered as a 2 sequence and 1 period study design. So this is mainly applicable to the drugs which are having long elimination of life. So elimination of life, it is more than 24 hours. So the problem, okay, the main drawbacks of this design is, okay, here we are comparing the plasma concentrations of test drug in a particular healthy volunteer is going to compare with the other particular volunteers. So there is a lot of intersubject variability come into the picture, because okay, it's not ideal to compare it is not ideal to compare the concentrations of test drug in one person with the concentration of the reference drug with the another person. It is not ideal situation. That is the reason parallel design is very rare in the bioequivalence studies. So okay, to get the preliminary data, sometimes if the drug is having long elimination of life, the people use it to do parallel design. People design to the parallel design. So coming to the crossover design. So here the healthy volunteers are again randomized, and they are divided into 2 groups. So like 7 subjects and here 7 subjects. So sequence 1, sequence 2. So here This design is called as a 2 sequence and 2 periods. We have 2 periods, 2 sequence and 2 period design. This is the most conventional design which is commonly used by the bioequivalence centers. So in this case, in the first group of volunteers will be administered a test drug in the period 1. So here they are going to stay for 3 days in the period 1. Like if the volunteers are coming on Monday, so they stay here Monday, Tuesday, Wednesday. So and then they are going to leave the bioequivalence center, and then they are going to take some break. So that is called washout period. So and then they are going to again visit the bioequivalence center on again Monday. So this is started on Monday. Again they may come to the period 2 on Monday. So we give one week, like 3-4 days washout period here. So usually the washout period is calculated based on the elimination of life again. So the company where I worked under the projects with what I have experienced, we need to give 10 elimination of lives as a washout period. So because when you give in the first case, when somebody is administered with the test drug, the concentrations remain in the body for a long time. So we need to before giving the reference drug, we need to make sure that there is no test drug in the body of the healthy volunteers. So for that we need to give washout period. Washout period is usually given as a 10 elimination of lives is considered as a washout period. So in this design, in the same group of subjects we are giving test drug and reference drug. So that means in the same volunteer is going to receive the test drug as well as reference drug. So both the drugs are given to the same volunteers. We can compare the concentrations of the test drug with the reference drug in the same volunteer, so that we can understand what is really happening. So how the pharmacokinetic variables are changing, we can clearly understand. So this will reduce the inter-subject variability, but not intra-subject variability. So we are not comparing with the concentrations of a particular volunteer with the concentrations of other volunteer. So we are comparing the same concentration of the test drug with the concentration of the reference drug in the same volunteer. So that is why there is no inter-subject variability in this design. But there is intra-subject, there may be intra-subject variability, but it's not tested here. So in this design is also considered as ideal design. Here the study period is more comfortable to all the bioequivalence centers. So that is the reason here the volunteer is not spending much time. Initially 3 days and another 3 days again. So the total length of the study is very reasonable and comfortable to the many centers. Unless this is commonly used for the drugs which are having less variability. So we can say when we have less variability, we don't have to see the intra-subject variability. So this design particularly useful for the drugs which are having less variability, and inter-subject variability we are already assessing, but intra-subject variability we are not assessing in this design. So coming to the replicative study design. So in this design, same pattern I have shown here, sequence 1 and sequence, it is sequence 2, this is a spelling mistake. And we see here, we have period 1, period 2, period 3 and period 4. We have four periods, 2 sequences. This design is called 2 sequence and 4 period design. So here we have a period 1, period 2, period 3 and period 4. We have 3 washout periods. So when you consider, this design is mainly considered with the drugs which are having high variability. So when you are testing the bioequivalence of highly variable drugs, highly variable from person to person, even within the person, maybe it is highly variable. So if it is highly variable within the person, then this is the best design we can use it, replicative study design. So you see here, the same group of subjects, we are dosing test drug 2 times and reference drug also 2 times. So we can compare the test this in the same person. We can test what are the concentrations here and here also. So if there is a variability within the subject, we can easily identify it. So this is a design where we can assess intra-subject variability and inter-subject variability both. So this design is most commonly used for highly variable drugs. Like when somebody is having for some drugs we have high first pass metabolism. Okay, such kind of drugs are more commonly used. Study designs in B studies. So like this is like a Latin square design. Like this is a 3x3 design. We can also say 3x3 design. So we have 3 sequences, like 3 group of healthy volunteers. We have 3 periods here. So in this design, the generic pharmaceutical companies, they are going to test two test drugs with one reference drug. So it is also possible. Okay, so we have only one reference drug here, but test drugs are 2 drugs. Okay, so in this drug, this model is used when we are testing more than 2 drugs. So Latin square design. So clinical endpoint study. So clinical endpoint study is like sometimes we don't have to estimate the concentrations, and we don't have to study the PK studies are not required. This consists of a randomized double-blind placebo-controlled and parallel design study comparing test product, reference product, and placebo product in patients. So we can conduct this one in the relevant patients also. So example, topical agents, nasal suspensions. Some people may ask, okay, how to conduct a bioequivalence study for topical agents like ointments. So those things are also here. We don't have to estimate the concentrations of the drug. So time versus plasma concentration graphs and all those things are not required here. So simply we compare. We see the therapeutic cure at the end of the study. So like by evaluating the difference between the proportion of the patients in the test and reference treatment groups who are considered a therapeutic cure at the end of the study. And then we also compare the superiority of the test and reference products with and compared to the placebo also. So these are the some of the like nasal suspensions, like topical solutions. We can use this study. So these designs already I discussed many of this. I would like to just compare this design and this design. Okay, here already I discussed the 4 sequence and 2 period design. So here 2 sequence and 4 period design. So in this case, so we have 4 groups of healthy volunteers, like we are following first sequence TT, second sequence like this. So in this case, we are different groups of volunteers are. In the first case, the same test drug is given to the first healthy volunteers. It is to test intra-subject variability. This is also to test intra-subject variability, but this is to test inter-subject variability. So because reference drug and test drug both are reference drug and test drug both are given to the same healthy volunteers. Okay, in this design, period 1 we have only 2 periods. So the time we spent is very less here. So this period is going to be completed in the less time. But whereas you see in this study, we are going to test the same healthy volunteers. Test drug is given twice and reference drug is given twice. So we can compare in this model, in this particular design, we can compare intra-subject variability and inter-subject variability in the same healthy volunteers. This is really wonderful. You can see in the sequence in the first group of healthy volunteers. Okay, test to reference, and test drug was again dosed here. So reference was given twice and test drug was given twice, and test can be compared with the reference also. So that is the reason. This is a design where in the same volunteers we can assess intra-subject and inter-subject variability both. But this it takes a long time to complete the study because we have 4 periods here. So we have 3 washout periods. It is difficult to get the volunteers get back to the bioequivalence center. Okay, then so what is the usefulness of the pilot studies? Pilot study is a small study where we like generally conducted in a small number of subjects, like 12 to 14. So pilot study is used to validate the following. Sometimes the sponsors they want to conduct a. So most of the time, okay, USFD, all the regulations, they will accept the pivotal studies. They will consider the data of pivotal studies, but not the pilot studies. Pilot studies are generally conducted to get some idea about the to get some idea about analytical methodology and PK variability, how the pharmacokinetic parameters are variable for this drug in the volunteers. Okay, they want to know how their formulation is going to vary with respect to PK parameters, and determine sample size to achieve adequate power. So sometimes they want to use the sample size so initially they may not know what is the sample size for pivotal studies. Sometimes we conduct a few pilot studies to know in order to know the sample size in the pivotal studies. So that's why pilot studies are useful to standardize the analytical methodology, and sometimes we can understand how pharmacokinetic variability of the formulation and determine the sample size. And sometimes we can optimize the sample collection time intervals, like at what time intervals we need to collect the blood samples, that can be finalized in a better way in pilot studies. So pilot studies are more useful to decide all these things. So general approaches for bioequivalence studies. So here I am just emphasizing the important points. I may not cover so many points, but however, these are some of the important points in the bioequivalence studies. The highest marketed strength should be administered as a single unit. So we need to, for bioequivalence, we need to test highest marketed strength. So like whatever the dose which is going to be marketed with the highest dose, that has to be tested. An adequate washout period, like 5 half-lives of the moieties to be measured, should separate each treatment. So regulation says 5 half-lives may be sufficient to give the washout period, but more than 5 half-lives are required. Usually the bioequivalence centers, they give 10 half-lives washout period between the periods, to make sure that when the first drug was completely eliminated from the body. And the test and reference products should be administered with about 250 milliliters of water to an appropriate number of subjects. So we see most of the time this much volume of water is given along with the test and reference products. So in the company where I worked, so when we give the drug to the healthy volunteers, we make sure that it is we just give the tablet to the healthy volunteer. We make sure that whether he has swallowed or not. So sometimes we test. We ask him to open the mouth, and whether we'll check it. Sometimes some volunteers will play the tricks. So that is the reason we make sure that so he has swallowed the tablet or not. Sometimes we don't allow them to go to the bathroom to spit it out. So some volunteers may intentionally do they spit out the tablet. So we just we have to make sure that whether the volunteer has swallowed the tablet to confirm it. And the blood sampling should continue for at least 3 or more terminal elimination half-lives of the drug to capture 90% of the AUC, relevant AUC. So here the blood fixing the blood sampling time point is more crucial. When you miss the time points which are very essential, so then it is a disaster. It is like a disaster of the study. So like I used to fix the time points in the company where I worked. So I used to take the very close points, just Tmax. Okay, either side of the Tmax, I used to take the close time points, and so we used to make sure that here they mentioned should continue for at least 3 or more elimination of half-lives of the drug to capture 90% of the relevant AUC. So many people get what is the last time point to be fixed. So we know that for complete elimination of the drug, it takes around 5 to 6 half-lives. So at least we need to multiply with the elimination of half-life with 3. So that could be the that it is better to take more than that time point. For example, I give an example. So if the elimination of half-life is 4 hours, so we have to multiply with the 3 as per the regulations. So 3 into 4, 12 hours. So we need to collect the blood samples till the 12 hours. But we usually go beyond that. So we are sometimes, okay, when we multiply the elimination of life with the 5, that is like, for example, elimination of life is 4 hours, 5 into 4, 20 hours. So last time point we collected 24 hours also. So depending on the elimination of life, we need to fix the time points. So we need to make sure that, okay, before Tmax there should be one or two time points. So that is also more important. Before Tmax we need to have, for example, Tmax is coming within half an hour. We need to make sure that, okay, there is at least 1 or 2 time points before 30 minutes. Sometimes we need to collect the blood sample within 15 minutes of the drug administration. So that's what we check it. And the other people may not know about okay, all these time points, because whoever writes the protocol here and there, they are going to discuss with the coordinator, study coordinator, and finally fix the time points. So as per the protocol, everybody follows the protocol. So the other people may not be knowing much about, okay, what are the rationality of these time points. But okay, mainly the protocol, the CRA and CRC, they are responsible for the proper fixation of the time points. So we need to make sure that, okay, 90% of the AUC should be captured in the time points. So for example, okay, in case of so for a drug, we need to collect the 24th hour sample, but we collected the samples up to 18th hour. So definitely the study is going to be collapsed, because we have not collected the blood sample. Okay. It should be like should cover minimum 90% of the element AUC. So we need to have around 3 to 4 time points in the elimination phase. That is also more important. So we need to have 3 to 4 elimination time points in the elimination phase, because we need to calculate slope and from that, okay, we calculate elimination rate constant and so many things. PK parameters are calculated using that. So okay, I'm just moving on. 3 or more samples should be obtained during the termination. That's what I mentioned here. During the termination log linear phase, to obtain an accurate estimate of terminal elimination rate constant. Okay, here I would like to tell one more point here. So I explained in parallel design sometimes. Okay, we need to in parallel design. Okay, for a drug which is having long elimination of life, we don't collect the blood samples till the end of the study. So we don't even collect the only in the first absorption phase, particularly in the first absorption half-life. In the first absorption half-life only we collect the blood samples. That is called truncated AUC. Truncated AUC is more applicable to parallel designs, and where the drug is having long half-life. So there we have no chance of collecting blood samples. Like we can't collect the blood samples for 7 days or 6 days. That is the reason we can collect the blood samples up to 3 days. So that means we need to make sure that, okay, first whatever the blood samples which are collected in the absorption phase, they are considered as the truncated AUC. Okay, general approach. I'm going to continue with the same general approaches. Usually a non-replicate randomized single dose and two period to sequence. Okay, don't have to be confused with the lengthy sentences like this. So I mentioned non-replicate, like, okay, normal crossover design. I mentioned non-replicate, randomized, single dose, two treatment like test and reference, 2 period means period 1 and period 2 sequence. So we have a TT, RR like sequence we have. So this crossover design is most commonly used in bioequivalence studies. A minimum number of 12 evaluable subjects will be included in any bioequivalence study. So this is most commonly in pilot studies. But in case of pivotal studies, the sample size is calculated based on the formula. So I don't have much idea about the sample size calculation in the pivotal studies, but so pilot studies are more useful to fix the sample size in the pivotal studies. So if the pre-dose value is greater than 5% of Cmax, the subject should be dropped out of the study. So it means here sometimes it happens that the healthy volunteer who is coming to the study, so who is going to recruited into the study already must have taken some same kind of drug in the in the body. Same kind of drug may be present in the body sometimes. That is the reason pre-dose value. It is like a pre-dose value before dosing the patient, the healthy volunteer should have the 0 concentrations of the drug. But in case if the pre-dose value is greater than 5% of the Cmax, so for example, Cmax is 100 micrograms. So if it is greater than 5% of this Cmax, like 5%, like if it is more than 5% of the concentrations are present in the body, then we can't include the healthy volunteers into the study. So this is what is the regulations are explaining it. So first we need to make sure that before including the volunteer into the study. So of course, we can't stop the volunteers to recruit into study, but study will be conducted. At the end of the study, we get the we get the concentrations. So then, when you see the data of the concentrations, when if there is a healthy volunteer who is having some concentrations at zero time point, that subject has to be excluded. Not the subject, actually the data has to be excluded. So it is not easy to exclude the subject here. So only thing is we are going to exclude the data, but not the subject, because we don't know what is the pre-dose concentration in the body unless we complete the study. So that is the one important thing here. Okay, so after getting the data, so when somebody is having pre-dose concentrations more than 5% of the Cmax, that healthy volunteer data must be excluded. Data from subjects who experience emesis during the course of a study or immediate release products be deleted. If vomiting occurs at or before 2 times of the median Tmax. Sometimes healthy volunteers, we have different studies like fasting study and fed study. So in case of fasting study, we don't give any food, but in case of fed study, we give defined food with high calories. So here, in case of fed study, we have to make sure that the patient is not vomiting. So even fasting study also, sometimes there is a chance that patient may vomit. Sorry for using the patient word. Okay, it is healthy volunteers every time. Okay, so if the healthy volunteers after taking the food, if they vomit, so not immediately after taking the food. So even generally 30 minutes after taking the food, the dose is administered. And the dose is, when the drug is administered, within 15 minutes or 20 minutes, sometimes the healthy volunteers may vomit. So such type of healthy volunteers, sometimes we will exclude them from the study, because during the study we will come to know already he has vomited. So this is also important concern. So this concern is different in case of immediate release products. This concern is like the vomiting occurs before 2 times of the median Tmax. Just 2 times of the for example, Tmax is coming at second hour, like before 2 times, like means it is like 1 hour before the Tmax. Okay, at or before the 2 times median Tmax. Okay, 2 times before the Tmax, if the healthy volunteer vomits, so we don't have to consider that healthy volunteers into the study. Okay, in case of modified release products, subjects who experience emesis at any time during the labeled dosing interval should not be included in the peak analysis. For modified release products, so they are like continuous. They are delivering the drug continuously. Okay, subjects who experience emesis at any time. So during the study, at any time, if the healthy volunteer is vomits, then we have to exclude the healthy volunteer from the study, or at least we have to exclude the data of the healthy volunteers. So we have in vivo bioequivalence, bioever. So for the certain drugs, we don't have to conduct the bioequivalence study. In vivo bioequivalence studies are not required. So we can do some in vitro experiments, and we can get the approval of bioequivalence. So it is called bioever. In vivo bioavailability and our bioequivalence studies may be waived not considered necessary for product approval. So for this particularly, BCS classification is more useful here to decide whether in vivo bioequivalence studies are required or not required. So here the dissolution test could be adopted as a surrogate basis for the decision as to whether the two pharmaceutical products are equivalent. In this case, see BCS class 1 high solubility and high variability drugs. So BCS class 1 drugs and BCS class 3 drugs also certain regulatory agencies they give the bio for these two type of BCS class 1 and class 3. So I have mentioned in the coming slides. So BCS class 1 highly soluble and highly permeable. So when it is solubilized very rapidly in the very rapidly in the GI fluids. Okay, it is unlikely that there is more variation between the generic product and innovative product. This is what they recommended. So that is the reason, like okay, we have some solutions. Solutions are coming under this category. They are highly rapidly soluble. So for such solutions, we don't need in vivo bioequivalence studies. So we can just go ahead with the bio. You can apply for bio. Pharmaceutical companies can apply for bio. So let us see more about bio. The drug should have high solubility and high variability according to BCS. And the requirements, so dissolution test in 3 different media. Like they have to conduct the dissolution test in the 3 different media, like buffer pH 1.2, buffer pH 4.5, buffer pH 6.8. So they have to prove this. Okay, it is having a high solubility in 3 different media. And 12 samples in each media, paddle rotating at 50 revolutions per minute. Okay, so we should follow all these norms, okay, as per the guidelines. The products are similar if the similarity factor is greater than or equal to 50, and both products show greater than or equal to 85% of dissolution in 15 minutes. So this is the important regulation. Okay, one has to pass. Okay, when there is any solution which can easily pass the bioequivalence study with the dissolution test. So I just defined highly soluble, highly permeable, and rapidly dissolving. So the regulations define different like a drug substance is considered highly soluble when the highest dose strength is soluble in less than 250 ml of water over a pH range of 1 to 7.5. The highest dose strength should be soluble in less than 250 ml of water. So that is considered as a highly soluble drug. Highly permeable drug. A drug substance is considered highly permeable when the extent of absorption in humans is determined to be greater than 90% of the administered drug. So if you have some literature stating that okay, greater than 90% of the drug is recovered in the urine. For example, okay, the drug is administered orally, and okay, the 90% of the drug is recovered in the urine. That means okay, it is the bioavailability is more than 90%. So it is considered as a highly permeable drug. So you don't have to do the bioavailability study also there. Okay, if you have a literature stating that okay, the drug is having more than 90% bioavailability, so it is called highly permeable. Okay, but the only thing is we need to prove this solubility test by dissolution and rapidly dissolving. So this is applicable for certain other conditions like I mentioned class 3 drugs, okay, where this is applicable. Immediate release drug product is considered rapidly dissolving when no less than 80% of the labeled amount of drug is substantially dissolved within 30 minutes. So 85% of the drug must be dissolved within 30 minutes of the time. Then it is considered as a rapidly dissolving. So these are the BCS and bioavers. There are certain guidelines, certain regulations which are mentioned here. This is EMA, European Medicine Agency guidelines, and this is FDA guidelines, and this is countries where WHO criteria is followed. So when you look at this here, European Medicine Agency guidelines, they are giving the bio-waiver to the BCS class 1 and BCS class 3 drugs. And here in case of FDA is giving waiver for BCS class 1 drugs. Maybe you can go through all this information later. So this is available. And this is in case of WHO, the countries where the WHO criteria is followed, which is class 1, class 2, and class 3 drugs are considered applicable for the application of biowaivers. So coming to ICH guidelines, like ICH topics, mainly the important guidelines in ICH are safety, efficacy, quality, and multidisciplinary. So in ICH, we have different guidelines, like okay, we have efficacy guidelines. The efficacy guidelines we have from E1 to E20. So E6 is the good, GCP is coming under E6. So E6 is a good clinical practice. So this is the important thing which is practiced in all the clinical trials and bioequivalence studies. So entire studies should be in compliance with the GCP guidelines. So it could be either with the ICH GCP guidelines or Indian GCP guidelines. Hello, sir. Hello. Okay, okay, I'm just moving on. So we have GCP guidelines is considered as E6 guidelines. So we for Indian guidelines, we have okay here ICMR guidelines and Schedule Y GCP guidelines for clinical research in India. So Schedule Y guidelines are most commonly used, and we have so many amendments in Schedule Y. So this is institutional review. Okay, I'm not covering all the GCP guidelines because it takes long time to cover. I have just differentiated Indian GCP guidelines and ICH GCP guidelines, where okay, there are certain variations between these two guidelines. But however, both the guidelines can be followed. Like okay, in India, Indian GCP guidelines are can be followed. But even many people will follow ICH GCP guidelines in most of the countries. So considering the time, I'm just moving on. Okay, so recent amendments, recent regulatory amendments in Schedule Y of the Drugs and Cosmetic Act. So these are the three amendments which were implemented in India. Serious adverse event reporting and compensation. This is Rule 122 DAB of Schedule Y. So in this case, earlier regulations are different, and now regulations are further changed. So we have another regulation, another amendment like permission to conduct clinical trials and ethics committee, registration and functioning. So these are the rules which are amended in the Schedule Y of the Drugs and Cosmetic Act. So I briefly mentioned here, okay, before, after amendment and before amendment, okay, what are the changes happened here? So mainly when serious adverse event happened in the healthy volunteers. So earlier only sponsor was okay. Sponsor is going to be 24. Okay, 24 hours reporting was limited by the sponsor to the sponsor by the investigator. So here the SAE serious adverse event must be reported to the licensing authority and okay, ethics committee. Okay, so we are going to within 24 hours of the occurrence of the event to the licensing authority, sponsor, and AC. Actually, clinical investigator must inform licensing authority, sponsor, and ethics committee. Hello, sir.
Okay. Siva Reddy garu. Yeah.
Okay, sir. Can I am going to finish within 5 minutes.
Yeah. Okay. Right. Thank you.
Okay. So I am about to complete 2 slides, I think so.
Right. 2 more.
Okay. So these are the some most of the three. They are the major 3 amendments which were happened in Schedule Y. So they talked more about how to report the serious adverse event to the licensing authority. So in this schedule, they mentioned they have given licensing authority may inspect the sponsor and CRO, okay, all the facilities. So in the old regulation, maybe they inspect only the CRO, but here they are going to respect the sponsor and BIB centers. Here in this rule 122D of the Schedule Y, prior registration. Okay, all ethics committees must register with the DCGI. It is mandatory with this schedule, this rule. So there is I mentioned there is a lot of impact of new amendments on the bioequivalence studies industry in India. So these are my acknowledgements. So I express my gratitude to my Guru Professor M.V. Nagabhushanam Garu for giving the opportunity to deliver a talk. I also thank Principal Dr. R. Govindarajan and the management of Hindu College of Pharmacy. Okay, and I thank my supervisor and my principal at the college and my friends. So lastly, my Guru Professor Akula Annapurna. Okay, without her, okay, I would not reach this position. Without her support, I would not reach this position. Okay, thank you. Sir?
Yeah, thank you very much, Siva Reddy Garu.
Okay, sir.
And there are a few questions from the students and from the participants across the country. And,
okay, sir.
Our Sitaram sir also wants to talk to you for a few seconds.
Okay, sir.
On this session. Sir, Sitaram Garu, just wish him and please complete in few seconds. Sir. And thank you very much, Siva Reddy Garu, for your informative session. And there are a lot of applause from all the viewers. And
thank
you very much, Siva Reddy Garu, for your informative session. And there are a lot of applause from.
Okay, can I start? Hello.
Sir.
Good
morning. Sir. Sir, how are you, sir?
Thank you. I'm happy to. Good morning, Dr. Siva Reddy Garu. Are you able to listen? I'm
yes,
fine,
sir, yes,
thank
sir.
you.
Sir,
you have given all important aspects of bioavailability and bioequivalence and good clinical practices. Well, this is very important because the government of India is going to encourage encourage to replace the all patent products with the generic products. So in this connection, the in place of patent drugs which are going to expire, their patency within few months or years.
Yes, sir.
Ultimately, the government wants to see that all the drugs are available to treatment of chronic diseases like hypertension, diabetes. The government wants to see that they are available at a cheaper price to all the regulatory authorities, to all the people. Not only the government, the insurance agencies, and especially the governments of USA by replacing the patent drugs with the generic drugs. So I wish that this knowledge will help our research scholars and students to come with new replacements with the present patent drugs. So thank you, sir. Thank you very much. Wish you good luck. Wish you good future in the coming years. Thank you.
Okay, sir. Thank you, sir. Sir, why is not audible, sir?
Good morning to all enthusiastic and scholastic participants of Pharma Fraternity. It's most useful educating webinar session by Dr. Siva Reddy Challa, research collaborator, Department of Cancer Biology and Pharmacology, University, Illinois College of Medicine at Peoria, USA. Today's speaker, Dr. Siva Reddy Challa has focused on latest regulatory guidelines for focus on clinical research in BA/BE studies and good clinical practices. And SAR also has covered general approaches for conducting bioequivalence studies and acceptance limits for Cmax, Tmax, AUC, and other issues which has to be taken into consideration while conducting the BA/BE studies. And SAR also has covered Hatch-Waxman Act and its significance in respect of the generic market, and the Patent Term Restoration Act of 1984. And there is a beautiful slide which is showing the organization chart, that is the flow of activities starting from sponsor and the involvement of all clinical research associates, and the role of the CRO, and the role of FDA and CDSCO and institutional ethical committee in one slide. Beautifully, Siva Reddy has presented. And also, sir has given a brief about the designs of BA/BE studies, something like parallel studies and crossover designs and replicative study designs, etc. And also the biowaivers based on the BCS classification of the drugs. It is a beautiful session which also described about the Schedule Y and a lot many other issues related to the significance of the BA/BE studies and the Orange Book. And it's my privilege to thank our management and our college chairman, Sri Jupudi Rangaraju Garu, and secretary and correspondent, Dr. Madhusudan Rao Garu, Principal Govindarajan, and Vice Principal, Dr. A. Sunita Garu, for their support in conduction of this webinar. I acknowledge the unwavering support received from all departmental HODs, the faculty and staff members in conducting this PRAJWALA, a series of international webinars conducted for the benefit of pharma fraternity. My thanks also go to the people who have given their precious time in organizing this webinar. In particular, I would like to acknowledge and thank G. Ramakrishna Garu for his technical and design process, and Y. Ratna Sindhu Madam for coordinating the inaugural talks, Dr. Ramaya and Nagarjun Reddy for introducing the speakers, and B. Brahmaya for question answer sessions, Sridhar Garu for technical support. And I am also thankful all the resource persons who have greatly helped us in organizing this conference. We appreciate all their time for the delegates who have actively participated for the last 4 days in the webinar series. Thank you, everyone. Thank you for giving me this opportunity. And today we are ending this morning session, webinar session. Afternoon at 2 p.m. we will assemble for the next webinar session for international labeling requirements for pharmaceuticals by Harshvardhan Gumadi from UK. Thank you all. Thank you, Siva Reddy, and I wish you all the best for your better career and better.
Okay, thank you, sir. Thank you.
Thank you.
Thank you. Thank you all.